Okay, so I want to go over this paper called Axonal Transport Enables Neuron-to-Neuron Propagation of the Human Coronavirus OC43. And I also want to go over this paper called Human Coronavirus Polyadenylated RNA Sequences in Cerebrospinal Fluid for Multiple Sclerosis Patients. So this is actually an extremely old paper. It came out in 1997. So this has been something that's been on the radar of neurologists for quite some time. But then I also want to go over this axonal transport enables neuron-to-neuron propagation of the human coronavirus OC43. So here, let's look at what coronavirus OC43 is. So this is one of the typical human coronaviruses. And let's see the Wikipedia article. So here's a photo of it, and it's a member of the species beta coronavirus 1, and it infects human and cattle. The infecting coronavirus is an enveloped positive sense single-stranded RNA virus which enters its host cells by binding to this N-acetyl 9-O-acetylneurominic acid receptor. And this is a common receptor that is on the surfaces of neurons, if I am remembering this correctly. So let me look. So it's a predominant sialic acid found in human cells and many mammalian cells, and other forms such as N-glycolyl neurominic acid may also occur in cells. And so the residue is negatively charged at physiological pH, so this is physiological pH levels, and it's found in complex with glycans on mucins and also glycoproteins found at the cell membrane. So just know that this is a receptor, or I should say this is something that is on the surfaces of cells that this human coronavirus OC43 can interact with. So if I'm remembering correctly, I believe this is the one that, I think this might be the most recent one that has jumped from bovines into humans, but I might be misremembering, so do not quote me on that. Yes, so comparison of the HCOV OC43 with the most closely related strain of beta coronavirus 1 species, the bovine coronavirus, indicated that they had a most recent common ancestor in the late 19th century, with several methods yielding most probable dates around 1890, leading to authors to speculate that an introduction of the former strain to human population might have caused the 1889 to 1890 flu pandemic. Okay, so HCOV OC43 likely originated in rodents. So, I mean, it would be a bovine coronavirus, right? So it's normally within cows and cattle, and all the related species of the cows and cattle, but it might have crossed into mice first and then from mice gone into human. However, they're not very, it's hard to know this stuff because this stuff happened in 1890. So let's look at this pandemic. So this 1889 to 1890 flu pandemic, also known as the Asiatic flu or the Russian flu, was a deadly influenza pandemic that killed about 1 million people worldwide. It was the last great pandemic of the 19th century, right? So the most reported effects of the pandemic took place October 1889 with recurrence in March, June, November. Okay, so basically it came in and it seemed to have been recurring, right? And it's still with us to this day. It is still with us to this day. So even though it came about in the 1890s, how do I phrase it? Well, granted, we are suspecting that this is related to the human coronavirus OC43, but it is still with us to this day. So whatever it did, it swept through the population, killed off the people who were vulnerable to this particular type of coronavirus, and then we would presume that everyone left was able to just continue living their lives. Granted, this keeps on circulating throughout the human population every single year. It's not going away. People get infected with this exact same coronavirus multiple times throughout their life. Same thing with the other human coronaviruses. So, and apparently, the genomic virological study said that it's tempting to speculate that the virus might have been not actually an influenza virus, but the human coronavirus OC43. Okay, so let us continue. So along with the involvement in previous infections The mucus associated with the mucous membranes, mouth, nose, GI, respiratory tract. This N-acetyl neurominic acid acts as the receptor for influenza virus, allowing attachment to the mucosal cells through hemagglutinin, which is the receptor protein on the influenza viruses, which is an early step in acquiring influenza infections. Granted, though, this human coronavirus OC43 is also able to do that as well. So now let's go back to this axonal transport enables neuron-to-neuron propagation of the human coronavirus OC43. So, and I'm not going to go through the whole paper, I'm just going to go through the abstract. So, human coronaviruses, also known as HCOVs, are recognized respiratory pathogens for which accumulating evidence indicates that in vulnerable patients, the infection can cause more severe pathologies. HCOVs are not always confined to the upper respiratory tract and can invade the central nervous system, or the CNS, under still unclear circumstances. The HCOV-induced neuropathologies in humans are difficult to diagnose early enough to allow therapeutic intervention. So this is one of the problems with these coronaviruses, is that the neurological symptoms associated with them seem to appear when it's already too late, right? So making use of our already described animal model of HCOV neuropathogenesis, we describe the route of neuropropagation from the nasal cavity to the olfactory bulb and piriform cortex, and then the brainstem. So let's look up the nasal cavity. You get the HCOV OC43 in there, and so this is the olfactory bulb. So here is your nasal cavity, and here is the olfactory bulb. So 1 second. Do you want out? Sorry about that. So you have your nasal cavity that has all these neurons that are associated with smelling, and then they all come up into this olfactory bulb, right? So the route of neuropropagation from the nasal cavity to the olfactory bulb, and then the piriform cortex. So what is the piriform cortex? Let's see if they have a good, oh, they do not have a good photo. So let me go to the images and let's see if tools, usage rights, labeled for reuse. You know what, I probably, okay, maybe in the olfaction Wikipedia page, they will have it. Olfactory bulb, the mitral cells. You know what, I can't find it. So it is just another section of the brain, and then it gets to the brainstem after that. So it gets into your olfactory bulb, it then goes into this piriform cortex, and I'm sorry, I'm not a neuroscientist, so I won't be able to tell you what exactly this is. It's just another part of the brain, and then it gets into the brainstem. So we identified neuron-to-neuron. Oh, and I should also mention that this might be why the woman, you know what? No, we'll keep on going. So we identified neuron-to-neuron propagation as one underlying mode of virus spreading in cell culture. So the cell cultures, they had a model, and this model is supposed to emulate the brain, and then we can see the virus spreading in the cell culture that is supposed to emulate what this brain model is doing. So our data demonstrate that both passive diffusion, so just through molecules randomly bumping around, passive diffusion could spread the virus from neuron to neuron. That both passive diffusion of released viral particles and axonal transport are valid propagation strategies used by the virus. So what is axonal transport? This is when in the axons of your neurons, which are the long stretches of your neurons, there is the ability for certain molecules to walk along the microtubules that are stretched through the axon. So there is a possibility that the viruses are actually hitching a ride on the axons of the neurons to get from one part of the body to a completely different part of the body. And since the neurons seem to have these receptors that coronaviruses seem to be very able to utilize, it makes sense, or it's very possible that the viruses are able to use this as a strategy to move from one side of the body to another. So, or a way for these coronaviruses to go from your nose all the way up into your brain, right? So, and then the ACE2 receptors, I believe, are also on a lot of neurons as well. So this would be allowing the neurons to uptake some coronavirus or SARS-CoV-2. So let's see. Our data demonstrate that both passive diffusion of released viral particles and axonal transport are valid propagation strategies used by the virus. We described for the first time the presence along axons of viral platforms whose static dynamism is reminiscent of viral assembly sites. We further revealed that HCOV-OC43 modes of propagation can be modulated by selective HCOV-OC3 proteins and axonal transport. So our work, therefore, identifies processes that may govern the severity and nature of HCOV-OC43 neuropathogenesis and will make possible the development of therapeutic strategies to prevent occurrence. Importance: coronaviruses may invade the central nervous system, disseminate and participate in the induction of neurological diseases. Their neuropathogenicity is being increasingly recognized in humans, and the presence and persistence of human coronaviruses in human brains have been proposed to cause long-term sequelae, so long-term sequel complications, even after you've been infected with that virus. And remember, you can be infected with coronaviruses multiple times throughout your life. So there are four endemic coronaviruses that people really only have immunity against them between 3 and 6 months, and then they are vulnerable again. Granted, we don't contract coronaviruses every 6 months because those coronaviruses you have to come into contact with that coronavirus. So even after someone's become vulnerable, they might not come into contact with that coronavirus for a significant period of time, and therefore they don't contract it until they come into contact again. So that might be, how do I phrase that? So yeah, let's continue and keep on going. So using our mouse model, relying on natural susceptibility to HCOV-OC43 and neuronal cell cultures, we have defined the most relevant path taken by HCOV-OC43 to access and spread to and within the central nervous system towards the brainstem and spinal cord, and studied in cell culture the underlying modes of intracellular propagation to better understand its neuropathogenesis. Our data suggests that axonal transport governs HCOV-OC43 egress in the central nervous system, leading to exacerbation of neuropathogenesis. Exploiting knowledge on neuroinvasion and dissemination will enhance our ability to control viral infection within the central nervous system, and it will shed light on underlying mechanisms of neuropathogenesis and uncover potential druggable molecular virus host interfaces. So if you want, you can read this article yourself by clicking the free full text here, and I will leave the citation in the description down below. So I'm going to pause, take a moment before I read the other abstract, but I just want to say that if the SARS-CoV-2 and other coronaviruses, but specifically SARS-CoV-2, if they have proteins that are able to interact with the monoamine oxidases or interact with some of the enzymes and modulate activity, that could be a way for the body or the brain to start producing, or I should say start dysregulating our monoamines or dysregulating our neurotransmitters, which could result in some of the delirium that many severe COVID-19 patients are experiencing right now. So it is possible that there will be a wave of people who have delirium or the inability to think properly. And now some people might argue that we already experience that right now, where a lot of people can't think properly anyway. Well, just put that on steroids. This is probably, I guess that's not the best terminology because we actually are using steroids to help heal COVID-19. But put that concept on steroids, where these people, a lot of people are going to have dysregulated neurotransmitter levels, and that might result in mass hysteria and mass psychosis, right? So this is something that we really need to consider and be mindful of. Yeah, so, and granted, that isn't the only problem. Another problem I see is these viruses might cause inflammation of the brain directly, right? So you might have T cell responses or responses that can recognize that the viruses are within the cell or within the neurons, and then your immune system might find a way to attack the neurons, and that attack is going to end up resulting in autoantibodies, right? Or you might generate antibodies accidentally against neuronal components, and these antibodies are then going to cause progressive pathologies because those antibodies are attacking the neurons. So that's one strategy that the virus could use to cause pathogenesis. Another problem that might rear its head is that the viruses could be mimicking some of the peptide hormones and or the receptors of the neurons. And so when your body tries to produce antibodies against the virus, they are inadvertently producing autoantibodies against the neurons as well. And so there really could be a laundry list of different neurological conditions that could be the result of this virus progressing through the population, and we just let it happen, right? So hopefully we can get our act together and try to suppress this before it becomes much worse. So let me go on to the next paper called the Human Coronavirus Polyadenylated RNA Sequences in Cerebrospinal Fluid from Multiple Sclerosis Patients. So even in 1997, we were discussing the possibility that these human coronaviruses are playing a role in neuropathologies. So for instance, in multiple sclerosis, which many people argue is an autoimmune disease against the myelin sheaths of the axons of the neurons, that is one of the symptomologies, or at least that is one of the biomarkers of multiple sclerosis, right? And so I think we should just continue with this. So the abstract, the human coronaviruses represented by the two prototype strains, HCOV OC43 and HCOV 229E. And so let's look up HCOV 229E. So it's a species of coronavirus which infects the humans and bats. It is an enveloped positive sense single-stranded RNA virus which enters its host cells by binding to the APN receptor. So this is the membrane alanine aminopeptidase, also known as the alanine aminopeptidase. Just know that this is another receptor on the surfaces of cells, and this is a peptidase, so it breaks apart some peptides. And in fact, let me look this up because so this is located in the small intestines and the renal microvillar membrane, also in other plasma membranes. In the small intestines, aminopeptidase N plays a role in the final digestion of peptides generated from hydrolysis of proteins by gastric acid and pancreatic proteases. So you have your pancreatic proteases that break up initially a lot of the proteins into these smaller peptides, and so the aminopeptidases are taking those peptides and breaking them down further into what I would presume are their amino acids. So yeah, so just know that this is in the intestines. It's also a receptor for one strain of the human coronavirus that is an important cause for the upper respiratory tract infections. So it appears as though this must also be in the... Oh, here we have an RNA expression pattern. So what I like about these RNA expression patterns is it can tell you what tissue types these receptors are located in. So here, the liver actually expresses a lot of it. The blood expresses, so white blood cells can express it as well. So perhaps your white blood cells themselves are vulnerable to these coronaviruses, or at least to the 229E coronavirus. Looks like the kidney, the fetal liver, the bone marrow. Sorry, I need to turn my head sideways to see this. So the adipocytes, so your fat cells, the pancreas, the pancreatic islets, which is what produces the insulin, these all produce significant levels of the cardiac smooth muscle, the cardiac myocytes. So yeah, all that seems to be producing enough of this alanine aminopeptidase for those cells to be presumably, what would the word be, vulnerable. Granted, there might be other enzymes that are necessary for the virus to actually get into the cell, so don't take this as don't take it as gospel that it can actually infect all the cells. Just know that those cells are expressing the receptor. So let's go back to this. So these are important in human respiratory. These are important human respiratory pathogens, also associated with necrotizing enterocolitis. So this would be a disease of the colon. And let's look up a photo. And it affects mostly the intestines of premature infants. The wall of the intestine is invaded by bacteria, which causes local infection and inflammation that can ultimately destroy the wall of the bowel. So this is a pro-inflammatory response, which ends up allowing the bacteria sometimes to start basically invading and causing necrosis of the intestines. Anyways, let's continue. So the two previous studies, one describing the electron microscopic observation of the donut-shaped particles resembling coronaviruses in a perivascular inflammatory lesion of brain tissue. So what is a perivascular inflammatory lesion? And let's look up Wikipedia. Let's look up what perivascular means. So a perivascular space, also known as the Virchow-Robin space, is a fluid-filled space surrounding certain blood vessels in several organs, including the brain. So this is a type of, let's get this photo up. I wish interstitial fluid in the perivascular space. So this would be an example of the perivascular space that is surrounding this blood vessel, okay? So this isn't everywhere, it's just in certain areas you can see this. So they had perivascular inflammatory lesions. So this perivascular space was getting inflamed and causing lesions of brain tissue. And this was taken at an autopsy from a multiple sclerosis patient. So in previous studies, they have seen that in a multiple sclerosis patient autopsy, they saw these lesions, and they in the microscope, in the electron microscope, they saw particles that resemble coronaviruses. So they weren't able to run, you know, these fancy genetic tests on them at the time. They could only see an image that looked similar to coronaviruses in this. So let's continue. Another one reporting the isolation of coronaviruses from the brains of 2 multiple sclerosis patients. So this suggested the possible association between coronaviruses and the human demyelinating diseases. We analyzed polyadenylated RNAs, so polyadenylated, so a lot of RNA in eukaryotes, and prokaryotes also do this, but for a different reason. In eukaryotes, which are cells that have nuclei, they will, when the DNA gets transcribed into RNA, messenger RNA, one of the ways that the mRNA gets processed is through the polyadenylation at the 5' end of the mRNA. So let me get a image of what's going on. So this is an mRNA. This is the structure of a typical human protein coding mRNA. And this mRNA, its future is to become translated into a protein. And so this is, in this coding sequence, this, from the start to the stop, this part is actually going to be translated from the mRNA into a protein, into a final protein, right? But this entire mRNA, when it gets transcribed from the DNA, needs to undergo certain processing. So one of the processing that it experiences is this 5' cap, and then another one is this poly A tail. And so this poly A tail will help signal to the eukaryotic cell that this is something that needs to be translated by the ribosome into a protein. So this coding sequence will end up not just existing as RNA, but it will actually come to fruition to bring about a protein. So polyadenylation is the addition of a poly-A tail. The poly-A tail consists of multiple adenosine monophosphates. Oh, in other words, it's a stretch of RNA that only has the adenine base. So remember, the DNA has A, T, C, and G as the bases that can make up the letters of DNA. RNA has A, U, C, and G. Wait, A, U, C, and G. Yes, and A, U, C, and G. Those can make up the code of the mRNA, but at the end of the mRNA, they add a bunch of A's as a signal for the cell to read and to understand what the heck is going on with this RNA. It's a way to differentiate between, say, a virus mRNA and the mRNA of the normal human cell. Granted, sometimes the virus is able to get its own poly-A tail as well. So keep that in mind. They analyzed polyadenylated RNAs extracted from the cerebrospinal fluid of 12 randomly selected multiple sclerosis patients and 10 patients with other neurological diseases. So medullary atrophy, Parkinson's disease, polyneuropathy, senile dementia, headache, and toxic polyneuropathy by reverse transcription and polymerase chain reaction, so RT-PCR, searching for the HCOV-OC43 and the HCOV-229E sequences. So by hybridization analysis of amplification products, we detected, and this is, hybridization analysis, I believe, is a type of fluorescence strategy to see if it's a fluorescence strategy, just keep that in mind. And they detected HCOV-OC43 polyadenylated RNAs in 10 specimens of patients with multiple sclerosis. So there were 20 randomly selected multiple sclerosis patients. So basically half of them had HCOV-OC43. Furthermore, we found positive hybridization signals for HCOV-OC43 in other neurological diseases except for the toxic polyneuropathy specimen. So they found HCOV-OC43 in half the multiple sclerosis patients, and they also found it in some of the other neurological conditions. And positivity for HCOV-229E was observed in 7 out of the 20 specimens of multiple sclerosis cerebrospinal fluid, 1 in the headache patient's cerebrospinal fluid, and the medullary atrophy specimen also resulted in a positive for HCOV-229E. So we're seeing that in the cerebrospinal fluid, these patients that have these neurodegenerative disorders, predominantly the multiple sclerosis patients, they are testing positive to a high degree for HCOV-OC43 and HCOV-229E. So, and remember, these are the polyadenylated mRNAs. So this would be, perhaps you don't really have that much of an active coronavirus at the time, but what is appearing to be very likely is that the nervous system, this is an immune privileged site. So this means the immune system, unfortunately, does not work as efficiently in the nervous system as it does elsewhere in the body. So other places of immune privileged sites are, say, the testicles, the eye, for instance. Regardless, these coronaviruses seem to be setting up shop in the nerves, or maybe they're not the nerves, maybe they're the glial cells or something within the cerebrospinal fluid that allows for the preservation and the production of detectable levels of the mRNA. And if you have detectable levels of the mRNA, that probably indicates that there is active translation going on. So translation meaning the production of these proteins of the virus, or at least maybe there's not enough of it to have a lot of viruses, but there is enough to detect the mRNA. So moreover, using a solid phase technique, we reported for the first time the sequence of cDNA fragments derived from RNA This is the complementary DNA fragments derived from RNA extracted from the cerebrospinal fluid of multiple sclerosis patients belonging to the open reading frame, which codes for HCOV OC43 nucleoprotein N. Furthermore, C complementary DNA sequences revealed the presence of mixed viral populations. Now, granted, this is just saying that in these patients, these neurodegenerative patients, they can see that there is detectable levels of human coronavirus in the cerebrospinal fluid. Now, granted, that doesn't say that the other patients didn't also have these coronaviruses in their cerebrospinal fluid. It could also be that the body fought it off, but then as a result, it started creating autoantibodies against proteins that are on the surfaces of the neurons, and then that is causing this this progressive degenerative disorder, destroying the myelin sheaths of the axons. So, yeah, how to phrase that? So it really could be that when it comes to SARS-CoV-2, we might unfortunately have to deal with an epidemic of multiple sclerosis, because SARS-CoV-2 seems to be much more severe. Yeah, much more severe than HCOV OC43. And if remember, HCOV OC43 might have killed about 1 million people when it went epidemic, or when it went pandemic for its first time. So the first time that it came into the human population, it might have caused a pandemic, killed a lot of people at the same time, and then we got some semblance of herd immunity, which ends up making it so that HCOV OC43 cannot really spread throughout the population like wildfire. It's more of a slow burn where people get infected with it over and over again throughout their lives as they lose antibodies. How to phrase that? So, moreover, it might be that SARS-CoV-2 is going to be like HCOV OC43 on steroids, where it sweeps through the entire population. It's going to be nearly impossible to stop. And then we're going to lose immunity over and over again, like what happened with HCOV OC43. And this might also result in a larger production of these neurological sequelae and other autoimmune sequelae. And we didn't stop it, right? We refused. We went with the herd immunity response, and now we are going to possibly suffer the consequences of these long-term complications as a result of it. Now, granted, perhaps our children will be better suited at fighting off SARS-CoV-2, like how eventually the human population ended up being culled and then was able to, with HCOV OC43, when that came into the human population, probably killed a bunch of people, everyone left, had at least a little bit more resistance against it, and then eventually we evolved as a result of that. And basically, our evolution resulted in a more resilient following generations that were able to fight HCOV OC43 more easily. Is this going to happen with SARS-CoV-2? Most likely, yes. If SARS-CoV-2 ends up becoming an endemic virus in the human population, we are going to end up better. But I should say the survivors are going to end up more resistant, possibly, and then eventually humanity will evolve to handle SARS-CoV-2. But are we willing to go through that bloodbath to have that happen? I am not. I think that it is better for us to fight back against these diseases than to just let them cull the human population over and over again. So, with that, I'm going to leave this video, or stop this video, because I think that is enough for these neurological symptoms. I think this is enough for now. So this is my third video of the day, so I think I'm pretty much done. So I will leave the I'll leave the reference, the citation in the description below if you want to read this for yourself. So thanks for watching, and hopefully I will see you tomorrow.